Professor JOANNA MASEL, thanks so much for coming on to Evolution Soup, all the way from Melbourne, Australia. You are Professor of Ecology and Evolutionary Biology, University of Arizona, researching the question of evolvability, that is, why evolution works the way it does. Just recently, you and your team released a study on perhaps one of the most incredible topics out there, the origin of the genetic code. Welcome to the show, JOANNA. It really is a fascinating subject and one that many people have wondered about. So what's it been like to finally be able to present your findings to the world?
Thank you so much for having me. It's a bit of a contrast to the kind of work I usually do. I often do very mathematical, very arcane, esoteric stuff that hardly anyone understands. And now we're working on something that really anyone with high school biology can relate to. For biology, it was really sort of the pinnacle of 20th century biology that we figured out how is it that DNA codes for proteins. But all of the major players ever since then have been wondering, how did that evolve? How did that come to be? Like there's this crazy complex code, you know, but there were no answers. There were only questions, there were theories, there were speculations. And we haven't solved it completely. We don't know exactly how it happened, but instead of just speculations, we now have data that really tethers speculations into reality. And I'd say the biggest reaction I'd get is from other scientists. This really is unusual as a question in that pretty much everyone has wondered about it, no matter what discipline they're in. So people from across incredibly diverse fields doing different things can all relate to this question.
Before we plunge billions of years back into deep time to discover the origins of the genetic code, let's just hear a little bit about your background. JOANNA, where did you grow up and what were the influences that pointed you in the direction of this incredible work that you're doing now?
So I grew up in Melbourne, Australia, where I'm recording this right now. I'm currently visiting my family in my family home. In high school, I trained for the International Math Olympiad, so I learned a lot of math as a teenager. But I didn't really want to be a mathematician. I wanted to apply it to something. And the Human Genome Project was in the process of being published. And it seemed like that was going to revolutionize everything in biology in a way that was going to leave a lot of mathematical questions. So essentially, strategically, I decided to major in genetics because of opportunity. And then I've been a mathematical biologist ever since. So I went to Oxford for my DPhil, where I actually moved a little bit away from genetics. I was interested in prions, which are nasty diseases, mad cow disease, that replicate without using any DNA or RNA, just proteins. So really starting from scratch as to thinking how replication, what it is and how it even works. And then I sort of found that I was somebody who understood both prions and evolutions. So in my postdoc, which was at Stanford, I was really inspired by this weird prion in yeast that helps add new amino acids to the ends of proteins during evolution. And it was a beautiful case study of how you get some larger rather than just small mutations during evolution. And it really inspired me to ask questions about evolvability using math. And the yeast prion system basically says how does a small protein become a large protein during evolution, since they all start out small. But that inspired me to ask, well, where do the original proteins, what about the birth of a whole protein rather than the birth of just a part of a protein? It's kind of a chicken and egg story. Most people think proteins come from duplicating old proteins and then they diverge and then you've got a new protein. But where do the old proteins come from? And it pushes it further and further back in time. Where did the first proteins come from? And there are two theories: Big Bang and all the proteins that ever were made in the primordial soup, and continuous creation, which is that new proteins are born out of junk DNA all the time. So when I started as a professor at the University of Arizona, having done all this mathematical modeling before, I really wanted to open up a much more data-oriented line of research. And there I looked at how it is that you get something from nothing, if you like. How do you get genes from non-genes? How do new genes come from DNA when there wasn't a gene there before? And so part of that, we were giving protein sequences ages. How long was it since they got, invented by evolution? And looking at how old genes are different to new genes. And we were very interested in very short times, but what we found as we kept pushing further back in time, that there were some trends that just kept going. And so that's how we got to the current work, that we ended up noticing some genes so ancient that some of them were born even before the genetic code was finished.
I think the best place to start in terms of understanding your work is to ask, what exactly is the genetic code?
All right, so our DNA is a double helix, and there's G's that pair with C's, and there's A's that pair with T's. So there are these 4 nucleotides, and if you read along one of the strands, you get some sequence of GITC. And it's not just us that read along it. Biology, the biological machinery of the cell reads along it, and it reads it as triplets: GIT, TAC, CCA. And those triplets, like just say GAA, gets read and translated into 20 amino acids. So 4 nucleotides go to 20 amino acids. So GAA goes to glutamate. And those proteins become the workhorses of the living cells. Proteins do all the work. So the genetic code is which triplet, like why GAA and glutamate? Why are those two connected? And it doesn't have to be. It's a sort of an arbitrary code at some level as to what goes with what, and that's what that we know as the genetic code. Sometimes people think about the genome as the code that codes for being human or being a mouse or being a fly. But that's the genome, that's not the code. The code is the way that it's the method by which it's decoded so that you get from DNA to protein.
Well, the genetic code is shared by almost every life form that has ever existed, from bacteria right up to human beings. However, the origin of this code has been of great mystery and subject to a lot of confusion. Joanna, what did you and your team do differently to discover how the genetic code got to where it is now?
Well, so firstly, the genetic code took a while to finish. There would have been a process. It wouldn't have always been as complex as it was now. And there would have been life throughout. So our thought was maybe, there would have been proteins built on primitive early genetic codes, and there'd be proteins built with the complete genetic code. And so the key was to find proteins that were so old that they were built, they were invented before the genetic code was finished. And our reasoning is that those proteins might still show signatures of what life was like with an incomplete genetic code. And we thought in particular that they would use more of the there are these 20 amino acids that all proteins are made of, and that these really old proteins would use more of the amino acids that were added first to the code, that were there when the protein was invented, and they'd use fewer of the amino acids that were added last, because when the protein were invented, there would have been 0 of those amino acids, and they only would have occasionally got added during the subsequent 4 billion years of evolution. So we needed to come up with some statistical methods to identify which proteins are 4 billion years old and which proteins are only 3 billion years old as a comparison group, because the 3 billion year old proteins are going to have normal use of the genetic code, and the 4 billion ones will have an informative, different use of the genetic code. So, yeah, the methods are a little complicated, but there have been real breakthroughs not done by us that really made this possible. So basically what you're looking for is proteins that exist in bacteria, and they also exist in something that looks a bit like bacteria, but it's not, called archaea, that are radically different organisms. They often live in very difficult environments like salt flats or hot springs or very extreme conditions. And it turns out these two types of organisms are so distant that they were only related 4 billion years ago. So we kind of draw a tree of the relationship of proteins, and we're looking for things that have this very, very, very distant long branch relationship between these two groups. And then we have to use information both from the gene and also from what we know about the species. And it's called gene tree species tree reconciliation. It's a very new technique of the last few years. And we have to use fancier mathematical models of understanding how proteins evolve and of reconstructing from current proteins what we think their ancestor looked like. So there were sort of statistical advances that had happened in the last 5-10 years that made this work possible. And then we put those together, found the 3 billion year old proteins, found the 4 billion year old proteins, and compared their amino acid use. And then the things that were used more or less in the 4 billion tells us what was early or late in the genetic code.
Looking back, there have been numerous attempts at deconstructing the genetic code and even trying to find the origin of life itself. Many people, of course, have heard of the famed Miller-Urey experiment of 1952. Joanna, what is your opinion of these experiments, and do you think these scientists were asking the right questions?
So first, the Miller-Urey experiment is really cool. But it's asking a different question, and also extremely interesting question, but a different question. It's asking, how do you get non-life from life? And the Miller-Urey experiment basically threw together a bunch of ingredients that are believed to have been around back before life, and throw in charge and this and that and see what can we make, what appears spontaneously. And they found, wow, you can get amino acids really easily. Like amino acids can just form spontaneously out of the raw ingredients that we think they had. And that's very encouraging in terms of an early role for amino acids in life. But that's a different question from the origin of the genetic code. So the genetic code is a very fancy set of machinery that would have evolved in organisms that were already alive. You don't go straight from non-life to the genetic code. You go from non-life, well, we don't know how it happened exactly, but you go from non-life to a lot of very primitive things that become more complex step by step. There's DNA and then there's this other thing, RNA. These organisms would have already had very fancy machinery for dealing with RNA. So, two different questions. But unfortunately, I think because they were all so long ago that one question, maybe 4 billion years ago sounds a lot similar to another question maybe 4.1 billion years ago, but 100 million years is a lot of time. I mean, we don't know the exact dates, but just as a point of illustration. So I think what happened is people were wrongly interpreting the Miller-Urey experiment, and they were arguing, in the absence of better evidence, I guess, that whatever was easy to make must have been early in the code, and whatever was hard to make in a Miller-Urey type experiment came late in the code. And so that was why they put, say, one amino acid is called histidine. And for a long time, people have assumed that histidine was late in the genetic code. And the reason is it's extremely hard to make histidine outside of living creatures. But histidine looks a lot like the purines that are the building blocks of RNA. So if you had a cell that was already alive, that didn't have a genetic code yet, didn't have long proteins yet, but it already had purines, then it would have been quite easy for that cell to make histidine. So indeed, what we found, in our work is that histidine was extremely early. And when you think about it, that's totally unsurprising because histidine is the most important amino acid in the active sites of key enzymes that are really ancient. So we found something that was different to what people thought, but when you look back, it's like, oh yeah, that answer does make more sense. Another example is the Miller-Urey experiment threw all these ingredients together, but it didn't add any sulfur. And sulfur is an element. If it's not there, you can't make anything that uses it. So two of the amino acids, methionine and cysteine, they have sulfur in it. So they weren't in the Miller-Urey experiment. They weren't made. And so but because they weren't in the Miller-Urey experiment, people thought they must have come late. But it turns out that methionine was also a really early amino acid. And indeed, we have a feeling that, in fact, there was a lot of sulfur in the early life environments.
Your team analyzed amino acid sequences across the tree of life, going all the way back to the last universal common ancestor, or LUCA, the shared ancestor of all life on Earth. Joanna, why was LUCA important to your work?
So we think of LUCA as the limit to how far the data can take us when we're looking at species. So just say you have sequences from different organisms across the tree of life, and they diverged a very long time ago, and you analyze them statistically. The limit to what you can learn about is the time when they all had a common ancestor before they diverged. And so by it's like triangulation. You look at what this sequence is like, what that sequence is like. Once they were the same sequence, you can kind of tell something about what that sequence used to be like that they both came from. But sequences even before that, you have no information about. Anything could have happened. There's no triangulation. You basically only have one source of information about the time when they first came apart that you were able to triangulate to get to. So when it comes to species, that is these bacteria and these archaea are the most distantly related species that we have. Some people might argue about where viruses fit in, but that hasn't been sorted out yet. So for now, it's bacteria and archaea. We ignore eukaryotes like us. They came much later by a bacterium and an archaea probably fusing in some way. So we say that time when bacteria and archaea separated, we call that the last universal common ancestor, or LUCA. And of course, if you think about the time in history when LUCA was around, there would have been lots of other life around too. But all of those other living things, except for LUCA, went extinct. Most species that have ever existed are extinct, and that includes all of LUCA's contemporaries. And so LUCA was the one that hit the evolutionary jackpot, that maybe it was better in some way, or maybe that was just chance. It might even have had something to do with the genetic code that it was so successful. And so it was so successful, it wiped all of its competitors out, and all species today trace their origins back to LUCA. But the interesting thing for us is, although LUCA is the limit of where you can go at the species level, we were interested in the origin of genes rather than the origin of species. So LUCA was a complicated cell that had already got some genes that had duplicated and diverged and were whole gene families, the different copies of genes that did different things. And so the common origin of those gene families is even older than LUCA. And so that allowed us, to push back inference and to start to see things even further back in time in LUCA, even though we came into this sort of thinking that LUCA was the limit to how far back you could still infer facts from data.
Well, it's very important work that you and your team are doing, Joanna, and I am very grateful that you've been able to take the time to come on to the show today to share your findings with us. Your work is certainly ongoing, so what does the future hold?
First, I want to say I don't think the work is important work. We're not improving health. We're not improving agriculture. The work is interesting. I mean, who doesn't want to know what happened 4 billion years ago? Not everything has to be important. Some of it is just interesting. It's just our curiosity. We just want to know. The genetic code is amazing. We want to know how on earth did that happen? So we're not doing it because it's important. We're doing it because it's interesting. And scientists often say that, and the general public mightn't see it. But I think with this one, I really noticed the general public does see it, that, yeah, we do want to know what happened 4 billion years ago. But in any case, this is definitely not the final word. We think there's a lot more we can get out of the data. One issue in the work we've done so far is that we only study the survivors. And so just like most species that ever lived are now extinct, most proteins that were ever part of life are also extinct. And there can be real bias as to which proteins go extinct and which proteins survive and become flourishing families of genes. And so one thing that's ongoing is we're studying those biases, which are very real and that might, potentially contaminate our work, although we don't think they did. And we're also developing new methods that'll get around that problem, that instead of just looking at a cohort of old genes versus slightly less old genes, we're developing methods that will tell us how a gene we had that is old has changed over time, how it might have gained the new amino acids and lost the old ones over time. So we're working on methods that will give us that resolution and get around any survivorship bias problem. And then hopefully it'll also tell us not just which amino acids were early and late, but also the sequence of events, which triplet changed from doing one thing to doing something else. Like what was the detailed series of events by which the genetic code came about? And also, I told you about these proteins we found that we get clues even older than LUCA. We really want to follow up a lot on them. We think we can push our ability to use data back further than ever before. Those we call them pre-LUCA gene families were really quite different from the LUCA ones, and there are all sorts of wild possibilities, about competing genetic codes transferring in, maybe even independent origins of life, all kinds of things. We have a source of incredibly ancient information, older than we dreamed was possible, that LUCA is not the limit. And we have new techniques that have really been coming out just in the last year or two about how to study things using protein structures rather than just protein sequences. And so we think there's a lot more we can learn from these sequences.
Joanna Masel, thank you very much indeed.
Thank you so much for having me and allowing me to share this exciting work.